Hi, I'm Allison Martin, and I'm a biochemistry student at SUNY Fredonia. I'm here today to give some highlights from one of the presentations I'm delivering at this year's SUNY Fredonia Research Expo. This poster was created as a structural analysis project for the advanced biochemistry class at Fredonia, and we used Accelerus Discovery Studio software to create the images you see here and do the analysis. The protein I examined here is called HRas and is also known as pg21. This protein is a critical intermediate in signal transduction, and I just want to run through a couple highlights from this poster. From the background, HRas is part of the MAP kinase ERK pathway. Mutations which deregulate this pathway lead to uncontrolled cellular proliferation in cancer. Because of this, HRas is known as an oncogene. And the HRas gene was one of the first oncogenes to be discovered in the 1960s, and mutant Ras proteins are found in 20 to 25% of all cancers and lead to very poor clinical prognosis. On the left here we have the gene sequence, and highlighted in green is the mRNA transcript regions, which is what will be translated to form the protein. Of interest here is that the high GC content in the gene sequence allows for very strong regulation at the transcription level, which is quite important for something that is an oncogene. On the right, we have a phylogeny tree from the BLAST results for this gene sequence, just to show the fact that it is highly conserved across multiple species. The protein sequence is even more conserved due to codon bias. Here's the amino acid sequence for HRas, and we have both the normal mature form as well as a few mutant forms. And the color coding that I'm using here in this slide will be maintained throughout. Orange will always highlight GTP binding regions, cyan is the effectors, and red is for the mutations. This enzyme is a small monomeric GTPase, so GTP will be hydrolyzed to GDP to turn off the protein, and then GDP will be exchanged for GTP to turn it back on. When it's on, it will signal to the next protein in the pathway, and when it's off, it should not signal. Accessory proteins associated with this are GAPs and GEFs, and these increase the activity of the protein by several orders of magnitude. Zooming into the active site, we see circled in pink here the glutamine at position 61, which is responsible for hydrolysis. The gamma phosphate of the GTP is positioned both by the GTP binding region and also by one of the two water molecules coordinated by the glutamine at 61. One water molecule will be used to orient the oxygens on the phosphate, and the other will be used to cleave it from the GTP. Mutations which disrupt the distance between Q61 and the gamma phosphate will effectively prevent hydrolysis from taking place. In wild-type Ras, when GTP is bound, a tyrosine in the effector region is also tethered to the gamma phosphate with a hydrogen bond. This positions the effector region so that it's ordered and able to interact with the next protein in the signaling cascade. After the gamma phosphate has been hydrolyzed and GDP is bound, the tyrosine is no longer tethered and the effector region becomes disordered and can no longer signal. This is a very clear distinction between on and off in the structure of this protein, which is important for the normal function. When we look at catalytically dead mutants, what we see are that mutated amino acids deform the structure. Here we have a valine at position 12 instead of the normal glycine, and this deforms the structure such that the glutamine at 61 is pushed too far away to hydrolyze the GTP. The tyrosine in the effector region is no longer tethered to the gamma phosphate, and so while it is still ordered and signaling, it cannot be turned off. Here in a different mutant, we have a lysine instead of the glutamine at position 61. And so it also cannot perform hydrolysis. This mutant is interesting in that the tyrosine is again tethered to the gamma phosphate, but since no hydrolysis can take place, there is no on-off functionality to the protein. So thanks for watching. A free PDF of this poster can be downloaded at tinyurl.com/pqm2rgc. So thanks for watching, and I hope you enjoyed it.